Welcome everybody in this session. I am Dr. Nasrin Sultana Lavli, Associate Professor and Head of the Department of Physiology, Moinamoti Medical College, Kumilla. Today we'll finish endocrine pancreas, inshallah. Yesterday, that is in the previous class, we have finished diabetes insipidus. What are the basic differences between diabetes mellitus and diabetes insipidus, and we have already started glucagon. Now, today we'll finish glucagon and somatostatin. These two hormone left. Today we'll finish these two hormone. Okay. Glucagon. Glucagon, like insulin, glucagon is a polypeptide hormone. Already I mentioned you, as like insulin, it is also protein hormone, that is polypeptide hormone, and glucagon also synthesized by pre-pro-glucagon, then pro-glucagon, then glucagon, like this way, like insulin. Okay, but function is always counter functions of insulin, মানে anti insulin action. Insulin হচ্ছে কি anti diabetic বলেছিলাম, anti diabetic, anabolic hormone, anti catabolic, anti ketogenic, anti lipolytic. But in case of glucagon, I have mentioned you that it is glycogenolytic hormone, gluconeogenic hormone. It is lipolytic hormone, it is ketogenic hormone, and also it has calorogenic action. Now, one by one, for written exam, if the question come like these functions of, write down or discuss the functions of glucagon, you have to write in your written exam khata like this way: on carbohydrate metabolism, see here, it always increase glycogenolysis. তাহলে কি হবে? It causes breakdown of glycogen to glucose. তাহলে increase glycogenolysis, that is anti function of insulin. ইনসুলিনের অপোজিট অ্যাকশনটা করবে, ঠিক আছে? তাহলে ইনসুলিন কি করতো? দুটো বড় বড় ফাংশনকে ইনহিবিট করতো। কি গ্লাইকোজেনোলাইসিস এন্ড গ্লুকোনিওজেনেসিস, এই দুটাকে কি করে? ইনসুলিন ইনহিবিট করে। বাট সি হিয়ার, গ্লুকাগন স্টিমুলেট, that is increase glycogenolysis and increase gluconeogenesis. So ultimately it increase hepatic glucose output. As you know that glucagon is a diabetogenic hormone, so of course it should to raise blood sugar level in blood, so increase hepatic glucose output, so increase blood sugar level, because it is a diabetogenic hormone. On fat metabolism, it causes increased lipolysis, whereas insulin is anti-lipolytic. That is anti-lipolytic means lipolysis korbe na, ki korbe? Lipogenesis kore insulin, but here see it increase lipolysis. So it increase lipolysis by which mechanism? Do you know this? In which mechanism lipolysis occur? Insulin inhibit which hormone or stimulate which hormone? And glucagon stimulate which hormone? Insulin always inhibit hormone sensitive lipase. Already I discussed in insulin chapter that insulin always inhibit hormone sensitive lipase but stimulate lipoprotein lipase. That's why it is anti-lipolytic. But glucagon anti-function of insulin, it stimulate lipoprotein hormone sensitive lipase, which is very essential for fat breakdown, that is lipid breakdown. So glucagon stimulate hormone sensitive lipase, which causes lipolysis. Okay. It increase free fatty acid level. Glucagon increase free fatty acid level. Of course, free fatty acid, if free fatty acid level increase in blood, so this free fatty acid causes increased beta oxidation and production of more and more acetyl-CoA, and ultimately more and more ketone body formation occurs and ketogenesis occurs. That is ketosis, ketogenesis, ketonemia occurs. Okay, that is free fatty acid is an essential component for formation of ketone body. So glucagon causes increased lipolysis by stimulating hormone sensitive lipase, and it increases free fatty acid, so more and more ketone body formation occurs and ultimately increase ketogenesis. You have to remember this point because these are very essential for your written and viva exam. Now, on protein metabolism. See here, on protein metabolism. It increased transport of amino acid into liver cell. It increased transport of amino acid into liver cell and increased utilization of amino acid for gluconeogenesis. Just now we said that glucagon causes gluconeogenesis. That is, glucagon always stimulate gluconeogenesis. So increased utilization of amino acid for gluconeogenesis and increased transport of amino acid into the liver cell. Now, already I mentioned you that glucagon has calorogenic action. That is, it increases hepatic deamination of amino acid. By this way, calorogenic action occurs. That is, it increases hepatic deamination of amino acid. On beta cells, what about on beta cells? It stimulates insulin secretion. Do you remember regulation of insulin secretion? Here, there we mentioned that glucagon always stimulates insulin secretion. Glucagon always stimulate insulin secretion. This is very important MCQ question. Glucagon stimulates insulin secretion. These are all about the functions, but on electrolytes. Also, you can write down here on electrolytes. What about on electrolytes? On electrolytes, insulin causes potassium influx into the cell. That is one in insulin functions of insulin on one electrolyte, that is potassium. It causes potassium influx of potassium into the cell. So what will do glucagon? Of course, potassium ki korbe? It always increases blood potassium level. That is, it decreases influx of potassium. So on electrolytes, you can write down here on electrolytes, that is on potassium. Potassium, it decreases influx of potassium. If it causes decrease influx of potassium, so more potassium persists in the blood level. So it increases blood potassium level. That is, glucagon increase blood potassium level by decrease influx of potassium into the cell. But whereas insulin causes influx of potassium, that is, it causes decreased blood potassium level in the blood. Insulin causes decreased blood potassium level by increasing potassium influx into the cell, but glucagon causes opposite action. So on electrolyte, you can write down here, you can add one point here, that it causes decreased potassium influx into the cell, so ultimately increase blood potassium level. Okay, this is about written exam functions of glucagon, how to write down in your exam khata. Okay, other functions of glucagon, you can write down this your written khata. It is also very important for your MCQ exam also. Other functions of glucagon: it increase strength of the heart, it increase blood flow to kidney, it increase bile secretion, it decrease gastric acid secretion. Several times this question came in your written exam, MCQ exam. Glucagon inhibits gastric acid secretion. This is very important point. Glucagon inhibits or decrease gastric acid secretion. But see here, everything is increased. Increased strength of the heart, increased blood flow to kidney, increased bile secretion, but it inhibits or decreases gastric acid secretion. This is very important MCQ question. But other functions, that is, all these functions are probably of minimal importance in the normal functions of the body. This is the minimal effect, but you have to memorize this thing for MCQ purpose. Glucagon increase strength of the heart, increase blood flow to kidney, increase bile secretion, but decrease gastric acid secretion. Okay, have you understood my point?
Yes, ma'am.
Glucagon receptors, like insulin receptors, glucagon also has its receptor. Of course, it is protein hormone. All protein hormone receptor always lies in the cell membrane, as you know that. But here, one thing, always remember this here, liver is one of the major target organ for glucagon. But for insulin, we know that insulin, major target organ for insulin, liver, adipose tissue, skeletal muscle. Okay, three things. But here for glucagon, liver is the one of the major target organ for glucagon. Glucagon mainly acts on on the liver. Glucagon binds to hepatic receptors in the cell membrane and stimulate adenylyl cyclase, and thus increase the intracellular concentration of cyclic AMP. That is, glucagon acts by, that is all protein hormone, as you know that all protein hormone acts by second messenger pathway, that is cyclic AMP pathway. Second messenger cyclic AMP system, glucagon functions. Okay, for mechanism of actions of glucagon occurs by second messenger cyclic AMP pathway, and major target organ is liver. Always remember this thing. Okay, now how glucagon, we mentioned that glucagon increase hepatic glucose output, that is, it is a diabetogenic hormone. It always increase blood sugar level, and it causes ketogenesis. How? What is the mechanism? What is the mechanism? See here, from your Ganong. I have taken this slide from Ganong. See here, glycogen. Glycogen, that is, it always stimulate glycogenolysis, that is breakdown of glycogen to glucose. See here, glycogen is converted to glucose 6-phosphate, and glucose 6 from glucose 6-phosphate it is converted to glucose. Then glucose is converted to, that is ultimate fate is pyruvate. But what glucagon do here? See here, glucagon activates cyclic AMP pathway, ultimately stimulate protein kinase A. These protein kinase A inhibit two things here. See here, this protein kinase A always inhibits in this pathway, that is conversion of fructose 6-phosphate to fructose 1,6-biphosphate. Here it inhibits, and also protein kinase A inhibits the conversion of phosphoenol pyruvate to pyruvate. These two steps inhibited by protein kinase A, which is stimulated by glucagon. That is, whenever glucagon bind with cell membrane glucagon, that is hepatocyte receptor, cyclic AMP system activated second messenger system stimulated protein kinase A. This protein kinase A inhibit these two enzymes here. So ultimately it stimulates glycogenolysis, that is, glycogen is converted to ultimately glucose. That is, it stimulates glycogenolysis to increase hepatic output of glucose. By this mechanism, glucagon increase glucose output from the liver. This is how glucose increase hepatic output of glucose. Okay, now ketogenesis. How glucagon causes ketogenesis? See here, glucagon activates hormone sensitive lipase. Already I mentioned you that glucagon activates or stimulates hormone sensitive lipase, whereas insulin inhibits hormone sensitive lipase but stimulate lipoprotein lipase. This is very, very important MCQ question. Several times this question came. Insulin always stimulate lipoprotein lipase but inhibit hormone sensitive lipase, whereas glucagon stimulate or activates hormone sensitive lipase, which is essential hormone for lipolysis. So glucagon activates hormone sensitive lipase in adipose tissue, increase fatty acid concentration in blood, and ultimately causes ketogenesis, that is breakdown of fat, and more fatty acid accumulation occurs in the blood. And if fatty acid concentration increase in the blood, increase beta oxidation, increase formation of acetyl CoA, and ultimately more and more ketone body formation occurs. And if increased ketone body formation occurs, that is ketogenesis, ketonemia, ketosis, this is occurs, that is ketogenesis. Also, glucagon inhibits storage of triglycerides in the liver. So, it increased fatty acid concentration in blood. Ultimately, ketogenesis occurs. This is the way of production of ketogenesis by glucagon. So, how glucagon increases hepatic output of glucose and how it creates ketogenesis, already we discussed this thing. Okay. Now, see the different comparison, the metabolic action of insulin and glucagon. Comparison of metabolic action. See here, this slide I have taken from South Asian Guidon. Second edition of South Asian Guidon, I have taken this slide. See here, insulin action and glucagon action. If you difference, see here, on muscle tissue, insulin causes increased glycogen synthesis, of course. as we know that it causes glycogenesis, stimulate glycogenesis. It of course increase amino acid uptake, increase glucose uptake, increase entry of potassium into the cell. This is the function of, these are the all functions of insulin, as we know that. Now glucagon action, see here, glycogenesis, no response. Increase amino acid uptake, no response on muscle tissue. And increase glucose uptake, no response. Increase potassium uptake into the cell by insulin, but glucagon, already I mentioned, it causes decreased potassium influx into the cell and ultimately increase blood potassium levels. See here, decreased potassium into the cell. Okay, now liver, it prevents glycogenolysis. Of course, as we know that insulin always stimulate glycogenesis and glycolysis, but it inhibits gluconeogenesis and glycogenolysis. So it of course decrease glycogenolysis, decrease gluconeogenesis. These two things are always inhibited by insulin, but it is anabolic hormone. So it is protein, causes protein synthesis. Insulin is anabolic hormone. It increases protein synthesis. But what about here? What about glucagon here? See, increase glycogenolysis. That is, it stimulate glycogenolysis. It stimulate gluconeogenesis, decrease protein synthesis. Of course, glucagon always causes decreased protein synthesis. Okay. Now, on adipose tissue, insulin causes increased fatty acid synthesis. Increased fatty acid synthesis, that is lipogenesis. As we know that insulin is anabolic hormone, it causes fatty acid synthesis, but decreased lipolysis, increased storage of triglycerides, increased storage of triglyceride. But opposite action of glucagon, see here, decreased fatty acid synthesis, that is lipolytic. It stimulates lipolysis and decreased storage of triglyceride. Already in the process of ketogenesis, we mentioned that it decreased storage of triglycerides, so more and more fatty acid accumulates in the blood and ultimately causes ketogenesis. These are the differences or comparison of the metabolic action of insulin and glucagon. This is very, very important written question and also for Viva exam. Please memorize this question very nicely, okay? Now, factors affecting glucagon, very, very important written question and also for MCQ exam. Factors affecting or factors regulating glucagon secretion, and you have to study factors regulating insulin secretion. Comparison of this, you have to study like this. What are the factors stimulate or inhibit insulin secretion? What are the factors stimulate or inhibit glucagon secretion? Then you will better understand this thing. See here, stimulators, amino acid, all amino acids, that is gluconeogenic amino acids, stimulate glucagon secretion. Cholecystokinin, gastrin stimulates glucagon secretion. Cortisol stimulate, exercise stimulate, infections, other stresses, beta adrenergic stimulators, theophylline, acetylcholine. These are the stimulators for glucagon secretion. What are the inhibitors? Glucose. But in here glucose means, what do you mean by glucose? Here hypoglycemia. Please write down in your khata. Hypoglycemia is stimulator for glucagon secretion, but hyperglycemia is inhibitor for glucagon secretion. Write down here, high glucose level. Inhibitors for inhibition, hyperglycemia is needed. That is glucose, that is high glucose level, inhibit glucagon secretion, but low blood sugar level, that is low glucose level, that is hypoglycemia, stimulator for glucagon secretion. Somatostatin inhibitors, secretin inhibitors, free fatty acid, ketone, insulin is inhibitors to glucagon, but whereas glucagon is stimulator for insulin secretion. Phenytoin inhibitors, alpha adrenergic stimulators, inhibitors, GABA is inhibitors to glucagon secretion. So you have to remember insulin, regulation of insulin secretion and regulation of glucagon secretion. What are the basic differences? Basic differences. Please write down everybody. Basic differences between insulin regulation and glucagon regulation. Please write down everybody in your khata about autonomic nervous system. Write down everybody about autonomic nervous system. In case of insulin regulation, parasympathetic stimulators to the insulin secretion. Parasympathetic nerves stimulators to insulin secretion. But sympathetic and parasympathetic both are stimulated to glucagon secretion. This is basic difference. Please write down everybody in your khata. In case of autonomic nervous system, parasympathetic nerves are stimulator for insulin secretion, but sympathetic and parasympathetic both are stimulator for glucagon secretion. Now, in case of GIT hormone, in case of insulin secretion, gastrin, secretin, write down everybody, gastrin, secretin, cholecystokinin, GIP, and GLP-1, that is glucagon-like peptide 1, all are stimulator for insulin secretion. Again, I am repeating, GIT hormone, gastrin, secretin, cholecystokinin, GIP, and GLP-1, that is glucagon-like peptide 1, all are stimulators for insulin secretion. But see here, gastrin and cholecystokinin are stimulators for glucagon secretion. Only gastrin and cholecystokinin are stimulators for glucagon secretion, but secretin is inhibitors to glucagon secretion. This is the difference between insulin regulation and glucagon regulation. Now, about blood. Blood sugar, high blood glucose level, amino acid, and free fatty acid all are stimulator to insulin secretion. Please write down everybody in your khata. About blood, increased blood sugar level, increased blood glucose level, increased free fatty acid level, and amino acid level stimulate insulin secretion. That is, high blood sugar, amino acid, and fatty acid stimulators to insulin secretion. But see here, high blood sugar level, that is high blood glucose level, always inhibitors to glucagon secretion. High blood glucose level, inhibitors to glucagon secretion. But low blood glucose level, that is hypoglycemia, is stimulators to glucagon secretion. Please, everybody, please, you have to write down this thing. High blood sugar level, that is hyperglycemia, is inhibitors to glucagon secretion, but hypoglycemia is stimulators to glucagon secretion. Free fatty acid and ketone. Free fatty acid and ketones are inhibitors to glucagon secretion. Free fatty acid and ketones are inhibitors to glucagon secretion. But whereas, as I mentioned, that free fatty acid and amino acid are stimulators to insulin secretion. Also, amino acid also stimulators for glucagon secretion. This is same. Amino acid in case of insulin and glucagon. Amino acid, always remember, in case of insulin and glucagon, amino acid are always stimulators to insulin and glucagon secretion. But free fatty acid is stimulated to insulin secretion, but inhibitors to glucagon secretion. Now, insulin. What about insulin? Excess insulin always inhibits insulin secretion. Please remember this point. Excess insulin always inhibits insulin secretion, but insulin is always inhibitors to glucagon secretion. Excess insulin inhibitors to insulin secretion from the beta cells of the pancreas, but insulin is inhibitors to glucagon secretion from the alpha cell or A cell of the pancreas, islets of Langerhans of pancreas. Now, what about glucagon? Glucagon is stimulators to insulin secretion. Glucagon is stimulators to insulin secretion. Now, GABA. See here, insulin and GABA, these are the inhibitors to glucagon secretion. Inhibitors to glucagon secretion. So, what are the main inhibitory things for glucagon secretion? High blood glucose level, about blood, high blood sugar level, free fatty acid, ketones, these are the inhibitors, and insulin, GABA, these are also inhibitors for glucagon secretion, and gastrointestinal hormone somatostatin is inhibitors of glucagon secretion. You have to remember this point very nicely, this thing. But in case of insulin, sympathetic nervous system inhibitors to insulin secretion. Somatostatin is inhibitors. Somatostatin is inhibitors for insulin secretion. Somatostatin is common for both hormone. Insulin is somatostatin is secretion is inhibition, causes inhibition of both insulin and glucagon secretion. This point you have to remember. Somatostatin, this protein hormone inhibits both insulin and glucagon secretion, both insulin and glucagon secretion. Okay, these are the key points, differences between the regulation of insulin secretion and glucagon secretion. Now, somatostatin, last one, somatostatin. It inhibits insulin and glucagon. As you know that somatostatin inhibits two hormones, insulin and glucagon. But in recent edition Genome, that is 26th edition Genome, another hormone they included, pancreatic polypeptide. It inhibits not only insulin glucagon, also it inhibits pancreatic polypeptide. Also in recent 26th edition Genome, it is mentioned as somatostatin also inhibits insulin glucagon and also pancreatic polypeptide. That is three hormones is inhibited by somatostatin. But in your textbook, it is mentioned as somatostatin inhibits insulin and glucagon. Only these two hormones. But recent edition Genome, it is mentioned as three hormone. Okay. Now, source of secretion of somatostatin. Somatostatin is secreted from hypothalamus, D cells or delta cells in islets of Langerhans of pancreas, and D cells in stomach and upper part of the small intestine. That is, ek kothay bolte pari from GIT. GIT theko secrete hoy. Tale kotha kotha theke? Hypothalamus, D cells or delta cells of islets of Langerhans and GIT bolte paro tumra. Tale source hocche tinta jayga theke. Now, chemistry and half life. Somatostatin is a polypeptide hormone. It is synthesized in two forms, namely somatostatin 14, that is within 14 amino acid, and somatostatin 28 with 28 amino acid. There are two types of somatostatin, somatostatin 14 and somatostatin 28. Functions are same. Functions are same. ঠিক আছে? Half-life somatostatin is 2 to 4 minutes, but according to your textbook guidance, it is mentioned as less than 3 minutes. No problem at all, 2 to 4 minutes or less than 3 minutes, same thing. Okay, synthesis. Somatostatin is synthesized from the precursor of prosomatostatin. Prosomatostatin. Metabolism. Somatostatin is degraded in liver and kidney. Somatostatin is metabolized in liver and kidney. Now, this is the last slide for today, and we'll finish, already we finished endocrine pancreas, actions of somatostatin. This is the last slide for today. See here, somatostatin acts within the islets of Langerhans and inhibits beta and alpha cell. As we know that it inhibits the secretion of both glucagon and insulin. It decreases, several times this question came in your MCQ exam. Somatostatin also inhibits motility, movement of stomach, duodenum, and gallbladder. So you have to remember this point. It decreases the motility of stomach, that is gastrointestinal motility. It decreases GI movement, gastrointestinal movement. That is, it decreases the motility of stomach, duodenum, and gallbladder. Ultimately, that is, it decreases the gastrointestinal motility. It reduces the secretion of gastrointestinal hormone, that is gastrin, cholecystokinin, GIP, VIP. Already we mentioned that it also inhibits pancreatic polypeptide. So gastrin, cholecystokinin, GIP, VIP, all hormones are inhibited by somatostatin. Hypothalamic somatostatin inhibits the secretion of growth hormone and thyroid stimulating hormone from anterior pituitary. That is why it is also called growth hormone inhibitory hormone, because chemical structure of somatostatin and growth hormone inhibitory hormone is same. That's why it is called growth hormone inhibitory hormone. It inhibits secretion of growth hormone and TSH from the anterior pituitary. This is very, very important MCQ question and also Viva question. So thank you everybody. Thank you very much. That's all from me today.